As a computational cancer biologist, I like to think of my work as a data musician. Just as a composer has an infinite number of notes, chords, and melodies at their disposal to create a unique piece of music, cancer cells have an incredible range of potential paths to differentiate and transform, as they can undergo various genetic and epigenetic changes, leading to an endless spectrum of cellular states and behaviors. Given the nearly infinite possibilities for a cancer cell to differentiate, our greatest struggle as researchers is the allocation of time, money, and biological replicates to prove theories in the literature worthy of clinical trial success. To counter the traditional limitations in statistical significance from low sample sizes, or a narrow focus from only exploring the mechanisms of action that we set out to look for, we are now developing a paradigm-changing software approach. polyMORPHOS uses patient RNA-seq data converted to metabolomic fluxes to predict the fate of AML patients with a systems-oriented approach, which will ultimately unlock tailored precision therapies. Additionally, we have SOURIS to apply this new functionality to ongoing biological pursuits. By stratifying patients based on phenotypic characteristics relevant to your project, SOURIS reveals metabolic states that lead to positive or adverse outcomes in the best course of intervention. With the early elucidation of mechanisms, researchers can allocate resources strategically. This new bridge between theoretical and tangible results will reshape cancer biology. I'm Nathaniel Pauly, a developer of bioinformatics solutions at the Cancer Research Center of Toulouse in CERN. Join me as we unlock the potential of metabolic data calibrated from a robust patient sample space using advanced machine learning to bring personalized cancer therapies and a brighter future. This is metabolism, re-envisioned in such a way that takes into account significant reaction activity between experimental conditions and patient cohorts. This is made possible by calibrating gene expression data to a structured human GEM metabolic model using flux balance algorithms such as IMAT to universally take into account all enzymatic pathway interactions. After reactions are sorted into their corresponding subsystems, cellular function can be directly inferred among treatments being studied across time. All other relevant genetic information is also provided in the reaction's corresponding metadata. This revelation of cellular function unlocks unexplored avenues in the realms of survival prediction from RNA sequencing data obtained at diagnosis. Our new software, Metabolism-Oriented Regression Predicting Human Overall Survival, or polyMORPHOS, takes advantage of this new data type. Considering that cancer is a dynamic illness with a metabolic landscape that changes across time, polyMORPHOS uses an XGBoost forest algorithm to iteratively select features, predicting survival and death for early, middle, and late stages of the illness. Instead of training the predictive model on the entire patient cohort, polyMORPHOS removes from consideration the patients deceased in the previous iteration. Therefore, our initial results tell us that the mechanisms of action relevant for day 150 survival are entirely different from those at days 300 and 450, and therefore it is a necessity that therapies and experiments should be uniquely designed based upon the changing metabolic landscape of the specimen in question. In order to apply this new insight of metabolic survival predictors to ongoing experimental projects, we now employ polyMORPHOS' sister software, Simulation of Universal Reactions in Silico, or SOURIS, the French word for mouse. Just like a laboratory mouse model, after hundreds of randomized training simulations within the BEAT AML2 patient cohort, Positive and negative SHAP values are generated, indicating survival and death respectively. They are compared among the expression of a phenotypic condition of the researcher's choice. In this case, to further study its anti-inflammatory properties in relation to AML, we will be using the expression of ENTPD1 gene in high, baseline, and low expression profiles. In the survival model constructed for day 250, this exchange of the histidoglutamate molecule into the cytosol indicates a predictor for death when the reaction is in the forward direction, as shown in violet, and favorable in the reverse direction in yellow. In respect to ENTPD1 expression, we clearly see that the favorable outcome related to this reaction is localized to the low ENTPD1 expression category, while baseline and high ENTPD1 increase patient risk in respect to the histidoglutamate exchange. With SOURI, we can now establish a threefold correlation among phenotypic condition, patient survival, and direct metabolic function. In contrast, with the typical comparison of the high versus low extremes, we now introduce 6 new degrees of freedom to more thoroughly analyze the effect of phenotype on survival outcome. With these newly introduced dimensions of analysis, we can now directly map the metabolic survival effect across a wide array of phenotypic conditions of AML. Here, favorable effect is designated in blue per each metabolic reaction, and death effect is shown in red. The tiles grouped together designate a shared metabolic characteristic among all the experimental conditions being compared. This newfound understanding of common causality allows us to transform previous chaos into order, paving the way for truth to emerge, bringing us closer to a victory in this race against fate.